Hello, good evening everybody and welcome to the planetarium. My name is Jochen Müller, I'll be the host tonight and I welcome you to Berlin Brains. Yeah! For all of you who are here for the first time maybe, let me introduce this event series to you. Berlin Brains is the joint event series of the Urania and its 5 partner organizations this year in our 10th anniversary season. These are the Bernstein Center for Computational Neurosciences, the Einstein Center for Neurosciences, the Excellence Cluster NeuroCure, the Sonderforschungsbereich 1315, and last but not least for today, the Max Delbrück Center for Molecular Medicine in the Helmholtz Association. Together, we present neuroscientists and their work on stage, not only on the Urania, as all of you know today, but also in other venues. For example, today, the Kinosaal of the Prenzlauer Berg Planetarium. And we do this because neuroscience is cool. And neuroscience is not something that you should be afraid of or that you should, it's not rocket science, it's just neuroscience. And so we talk about the brain, its cells and functions in such a way that everybody will understand. The speakers really put some effort in preparing their preparations. We work with them hard on that. And so they really have the wish to present it to you, to get in contact with you. And because in the end, the work they do, scientists, right? They do this for us. It's demanding, it's a stressful work. They do this for us, but however, they can't always work with us. Especially today is an example for that. But today is also an example how they're still able to draw conclusions that are of high relevance for us, making our life better, hopefully, in the end. So today, Hanna Hörnberg and Tobias Kurth from the Laboratory for Molecular and Cellular Basis of Behavior from the Max Delbrück Center will talk about sex and gender in mental health and why sex matters. They show how they investigate depression in mice and how this is and why this is relevant for us humans. And they do this together because the conceptual motto of this event series is science is teamwork. Here always at least 2 people go on stage, mostly a PI or professor and a PhD student or postdoc. Today the PI is Hanna. She will give an introduction into the field of research she's working on with her group. And Tobias, the postdoc, will then afterwards give you a bit of insight about one concrete project of his. Together we will talk roughly about 45 minutes and afterwards we will have again another 45 minutes roughly for an interesting discussion with you guys. And when we are at the Q&A, I will just ask you to, who has a question, raise your arm and then I will come you with a microphone. Because, as you see there, say hi to Felix. Hi, Felix. We record these events also on video. So we have super interesting talks here 5 times or 6 times a year. Record them so that you can see all of them, even though you found out about Berlin Brains just today. Oh my God, you missed 9 seasons. Half of that you can see on the web. What did we have, for example, this year? We had in March how the brain controls appetite. In April, how the brain discriminates important from unimportant info in hearing. We heard some new infos on fine-tuning of brain stimulation in September. In October, we talked about the role of the eye movement in attention and to awareness. And today, the last event for this year is sex and mental health. Okay, video is cool. You can find them on YouTube or on urania.de. But of course, live is better. So thank you for coming here. And if you see the video, get your ass up, get to Berlin, get to the Urania or the Planetarium or wherever we do this show, because life is always better. Get in contact with us, talk to us, discuss with us. Today, I'll also ask you during the talk, or not I, Hanna will do this, to do something that we usually don't do in Berlin Brains. Get out your mobile phones. You can do this now. Check the sidebar. If it's loudless, then you did it right. But later on, you can use that. You can prepare it now. If you have the app Slido installed, open it. If you don't have it, you don't need to install anything. Just make sure that your browser is open and you're maybe already on the website, www.slido.com, S-L-I-D-O.com. and then you'll see it's a simple site, looks a bit like Google, just white with a field where you can enter stuff, and then we will tell you 4 letters, I think, that you can enter later on, yeah? And then you'll see what happens. Okay, and you can put them away now. Thank you. Wonderful, works well, better than with my son. Anyway, okay. So they will speak after each other. There will no be, I will not come on stage between them. But now let's introduce me to the 2 speakers. Hanna, she is from Sweden. And she studied biology in Uppsala before she went to Cambridge to do her PhD there in neurosciences. She then went to Switzerland, did her first postdoc in Basel, and in 2020, I think, she came to Berlin, and since then she's junior group leader at the MDC, where she focuses on, well, investigating the mechanisms of mental health disorders like depression. She will talk about that today. Tobias, he's from the south of Germany. He studied biology and bioinformatics in Munich. He then went to Regensburg to do his PhD then, switched to Atlanta, or Alabama, Atlanta, I always mix it up. Anyway, United States, came back, and and then came to Berlin in 2021 to join the research group with Hanna, and he started his postdoc here, and he focuses on, well, investigating the mechanisms of resilience in depression. Okay, they will talk together, and I now hope that we all together give them a warm welcome. Please, with a big round of applause. Hanna Hörnberg.
Great, so hi everyone, and really a big welcome from me as well. I'm really glad that so many of you come out today and hear about our research. So Jochen already introduced me a little bit more than I had expected, so you will hear some things twice now, but you just have to live with it. So so as I said, my name is Hanna Hörnberg. I'm originally from Sweden, so I'm from Stockholm originally. And the picture here is it's me, although you can't really see it, and my 2 pet rats as a kid. So they were called Zorro and Tintin. And I've always really loved animals, and I've always been very interested in behavior. So why we behave the way we do, and why we behave so differently. And I'm not from an academic family, so I never really considered studying behavior professionally. But one day in high school, we had visiting, I think, a PhD student from a university who was a biologist. And I thought, okay, this sounds really interesting. This is what I want to do. So I applied to do this at university. And so I went to Uppsala, which is a little bit north of Stockholm, with the idea that I was going to study zoology. So I wanted to study, in particular, whale behavior. But already in the first year, we had a course in cell biology and then another course in genetics. And I found this much more interesting. So I switched gear and went to more kind of molecular neuroscience. And eventually I did my master's studies in neuroscience. And for my PhD then, I wanted to study how the brain develops. And for that I went to England, where I studied early neural development in another model system, which is a small fish called zebrafish that is mostly see-through, so it's very easy to study kind of early developmental processes. But I did really miss behavioral neuroscience. So eventually then for my postdoc, I went to Switzerland in Basel, where I moved to study social behavior in mouse models. And I was particularly studying how social behavior is regulated in the brain using mouse models for autism. And then about 4 years ago, I was lucky enough to be able to move here to Berlin to start my own group in the Max Delbruck Center for Molecular Medicine. And we're located in the Buch campus, which is very north of Berlin. So at the moment, a really terrible place to be, since the S2 is not running. And I'm very lucky there to have a really fantastic team of people who are working with me on these research questions. And some of them are in the audience today, and you'll of course hear from Tobias later. So in my group, we're interested in understanding then the cellular and molecular mechanisms that regulates behavior. And we're particularly interested in the types of behaviors that are often altered in neurodevelopmental and psychiatric conditions, such as, for example, then depression, which is the topic of today. And the main questions we're interested in is understanding why some people are more likely to experience mental health condition compared to others, and also then how sex and sex-specific factors influence the risk of mental health conditions. And just before I move on, I just want to talk briefly about what I mean when I talk about sex and when we talk about gender. So when I talk about sex today, I will only really talk about biological characteristics. So often this is, of course, your chromosomes. But for us, actually what is most important is your sex hormones. So whether or not you primarily have female sex hormones or male sex hormones. And when I talk about gender, I mean it as just a pure social construct. And this will be very different depending on where you live and maybe how your family view these topics. And it's incredibly important for mental health and depression. But it's completely impossible to study in mouse models. So for most of the talk, we will only really focus about biological sex. So as we said, we study this in mice. And how can we study such complex human disorders in mouse models? So a mouse can't really recapitulate the entire of a human disorder. So a mouse cannot be depressed, or at least we don't think so. But they can be used to answer very specific questions about the biology of part of these disorders in ways that wouldn't really be possible to do in humans. And the reason we specifically can use mice is that they're very genetically similar to us, so about 93% similar. And they're also very biological similar, and in particular, like the brain circuits that are important for how we experience stress and anxiety, they're really similar in mice and humans. And what's also very important for us is that they're relatively small and we can control their environment completely, and they have a quite short lifespan. So we can study them from birth to adulthood in just a few months. And this, of course, would take much longer to do in people. So we can much more quickly see how, for example, events that happen to them during their childhood affects their later on behavior. So what mice allows us to do is really examine then how, for example, early life stressful experience or the personality of the mice, because mice do have very distinct individual personalities, how this affects brain development and ultimately how they will respond to, for example, risk factors for depression, either genetic ones or environmental ones. And then we can really study how these risk factors affect the brain on a cellular level, so really going to a single neuron. And this, of course, again, is not possible to do in humans. So, okay, I will move on to the topic of the evening, which is depression. So this is only part of what my lab does, but yeah, it's one of the really exciting parts, so I'm glad you're here to hear about it. So what is depression? Most of you probably know someone with depression or have experienced depression personally, and this is because it's very common. So almost 1 in 5 people will experience depression in their lifetime. And it's also almost twice as common in women as in men. It's also highly variable, so people can have quite different symptoms and also different prognosis and response to treatment. So 2 people with depression will not necessarily at all have the same experience. It's also episodic, so this means that it can come and go. So you might experience a period of depression, then become better, then experience another period of depression. And almost 80% of people who are diagnosed with depression will experience at least one episode or more than one episode during their lifetime. But of course, depression is also extremely personal, so it can have a really big impact on you and your friends and loved ones. So in order to see get a kind of a feeling of why you're here and what you think about when you think about depression, then please either scan this or put in the code so we can get a feeling of what depression means to you. So I have to say, being from Stockholm, the Berlin winters is like 100 times worse. So we have quite a lot of different things. Yeah, so as we can see from this nice word cloud, there's clearly a lot of different ways to think about depression, and a lot of ways depression can mean to you and how it can affect you. So obviously things like numbness, lack of joy, and sadness is kind of the key aspect of depression. Also isolation, it's very common that people isolate themselves. No motivation, tired, trauma, it's very highly linked to trauma. So I think what we have here is a really good overview of what depression is and what it can mean to be depressed, and what it can also mean to have close ones who experience depression. And this is also quite similar to what, I'm sorry, the colors are not really coming out here. So it's quite similar to what depression also is clinically. So in order to get diagnosed with depression, you need to have at least 9 of these clinical symptoms, at least 5 of these 9 clinical symptoms, and the first 2 are mandatory. So you have to experience depressed mood, and you have to experience anhedonia, which is a change in how you experience pleasure. But otherwise, the symptoms can vary somewhat. And what's also very important is that you have to experience a change in depressive-like symptoms for at least 2 weeks. So it's not enough that you just have kind of one bad day. You have to feel bad for a prolonged period of time in order to get a diagnosis. But because depression is so variable, not 2 people are necessarily the same. So although everyone has to have the first 2 symptoms, they can really differ both in the severity you experience symptoms, as well as which other symptoms you have. So some people might really have major problem with energy and lack of sleep, whereas others might have more problem with difficulty to concentrate, for example. This can also mean that depression can be very different in terms of severity. So some people might not be able to leave their bed, basically, or perform any daily tasks, whereas others might seem like they're functioning quite normally, but inside they're really not feeling well. So what are the risk factors then for depression? So I put up the kind of 3 main things here, which are also the 3 main things that can affect the brain. And one of them is genes. So we know that there's a large genetic component to depression, and it's not a single gene disorder, so there's not like one gene that's linked to depression. Instead, there will be variations in hundreds of genes that each convey a slight increased risk or a protection to experience depression. And then everyone have a combination of these genes, which in total will mean that you might have a slight increased risk or a slight protection for depression. Another important risk factor is the development. So this is both things that happens before you're born, but also things that happens during the kind of critical stages of brain development, so during childhood and your teenage years, so adolescence. And it's known that traumatic or stressful events during childhood or adolescence is one of the strongest risk factors for developing depression later on in life. And this then also ties into environment. So stress and trauma are the biggest environmental risk factors for depression, but also things like drug and alcohol abuse, or things that is really difficult for us to do anything about, like our socioeconomic background. So having economic stress is a risk factor for depression, which is perhaps not so surprising. So there's really multiple different factors that are linked to depression, which probably means that in the brain there's not really one depressed brain, like there's not one depressed person. And this, of course, then also has some effect on how we will think about treatment. Because it might mean that not one treatment will fit everyone. So what happens in the brain then when we are depressed? Because everyone, again, is so different, people's brains will not look the same. So none of our brains will look exactly the same, and 2 people's with depression's brain will also not look exactly the same. So what I'm talking about now are kind of big general changes. And one thing we know happens is that there are a lot of both structural and functional changes that happens in the brain when you're depressed. So the brain is basically a large interconnected network. And we know that some of these network or some of these connections will strengthen during depression and others will weaken. So for example, the networks that are important for responding to anxiety and stress, they might strengthen so that we over-respond constantly to stressful events that we normally would not find so stressful. And circuits that are important for cognition might instead weaken so that we find it more difficult to learn new things or kind of cope with our everyday life. And if we zoom in to where these connections are being made in more detail, So neurons communicate via structures called synapses. And these structures are not static, so they're plastic in that they might change in strength and in number. And this is really important for learning and memory. So it's absolutely necessary that we have this plasticity in synapses in order to learn new things and adapt to our environment. And in depression, this process is changed to the worse. So it's thought that it affects plasticity negatively, so that we might not be able to make as many new synapses or change our synapses in the same way. And this will then make it more difficult to us to learn new things or sort of adapt to things, to our environment. Neurons also communicate via neurotransmitters. So they're released from one neuron to the other neurons, and this is how kind of neurons send messages to each other and how information is conveyed throughout the brain. And we have lots of different neurotransmitters, and in depression, some of these neurotransmitters might increase and some might decrease. And this might happen everywhere in the brain, or it might happen like locally, just in parts of the brain. And this is also the mechanisms that are most commonly targeted with treatment. So the kind of gold standard treatment for depression is serotonin reuptake inhibitors, or SSRIs. And they work by increasing the level of the neurotransmitter serotonin in the brain. And it's thought that this eventually leads to kind of a remodeling of circuits that are important for mood. So serotonin it does lots of things, but one of the things it does is that it regulates mood. So it's thought that if we increase the level of serotonin, even if this is not the cause for depression, it might help to kind of reset the brain to work more like it did before you were depressed. But this takes time, and this is why it takes weeks for these to work. Although I should say we don't fully understand exactly how they work. But they do work, but unfortunately only in about 50% of people. So roughly half of people who are diagnosed with depression will not respond adequately to treatment. And we kind of don't know why that is. So we have no way to tell at the moment who will respond well and who will not respond well, or which antidepressant will work best in one person versus another. But one thing that might really play an impact in this is that there's kind of a lot of evidence that suggests that biological sex can influence how we respond to antidepressant treatment. And this is perhaps not surprising, given that sex and gender as a biological factor plays a really important role in mental health. So the graph I'm showing you here is a summary of a lot of mental health conditions. And on your right axis, so it's split into axes, so you have the right and the left. And based on what I've said, you could probably guess which axis is women and which is men. And someone guess? Yeah. So on the right side, we have women, and on the left side, we have men. So depression and anxiety disorders, which is very much linked to depression, are both in red. And as you hopefully can see, they're very much shifted to the female side. And this is true for all age groups. So women are much more likely to be diagnosed with depression from the age of 5 to 95. So that's fun for us. So not only are women diagnosed more often with depression, the symptoms of depression are also slightly different in men compared to women. So, for example, women are much more likely to experience changes in sleep and appetite, for example, whereas men are more likely to have outward symptoms like anger or increased risk-taking. And men are also much more likely to, or 3 times more likely to, die from suicide than women. So this difference in diagnosis and symptoms, it's not something we see in depression only. In fact, it's something we see in basically all disorders that affect the brain. So, for example, women are more likely to get Alzheimer's disease, so it's also fun for us, versus men are more likely to get Parkinson's disease. And there can be lots of different reasons for this. So, for sure, for example, our lived environment is really important for this, so our experience. So being a woman and a man in today's society is not the same. We're not treated the same necessarily. We don't have the same experiences necessarily. And this will impact both maybe your likelihood to get diagnosis, but also your willingness to go and get a diagnosis and get help. But because we see this in basically every disorder, it's very likely that there's also a biological component to this. But kind of my main point with this is that we don't actually know why there's a sex difference for any of these conditions. And one of the reasons we don't know this is because historically we have not really taken sex into account at all when we study neuroscience, or basically in any biomedical research. So despite the fact that we see this clear sex and gender difference in diagnosis for all these conditions, males are still pretty much the norm in clinical and preclinical research. So they have been the norm, and this hasn't really changed as much as we would want to. So the graph I'm showing you here is a summary of papers that are published in my field, so neuroscience, from 2009 and 2019. And in blue, you have papers that uses males. In the tiny bar in orange is females, and the black one uses both sexes, and gray they didn't even mention. So as you can see, the number of papers that uses both sexes have really increased a lot in 10 years. So that's good, right? No. Well, it's better than nothing. But most of these papers don't do any sex-specific analysis. And the number of these papers that do sex-specific analysis have not actually increased at all. If anything, it has slightly gone down. So this means that despite the fact that there are more papers that uses both sexes, this hasn't increased our knowledge at all about female-specific biology. So, sorry. And I mean, if anything, the papers that use only females have slightly decreased. So why does this matter? Like, is it really so important to care about sex? So this matters because we know that neglecting sex differences in clinical and preclinical research can have really negative health implications. And one of the reasons for this, or just one example, is that men and women can probably differ, or they differ in drug absorption rate. So for example, this means that if me and Tobias would take a pill, we might not metabolize it in the same way. So even if we were the same height and weight, we might get very different drugs into our system. And it might take longer, or it might stay longer. And this means that the dose we get in the end will be very different. And this is probably the reason why women are much more likely to experience adverse drug side effects than men are, including very serious ones that requires hospitalizations. There's also kind of no sex-specific treatments, medical treatments that exist, except for sex-specific disorders. And how kind of sex and gender-specific medical factors, like hormonal contraceptives that are incredibly common, and also sex hormones, how they impact treatment response in depression, and also how they just impact symptoms and our experience, is extremely poorly understood. So I think to give the right treatment to the right person, both in order to improve the treatment response and reduce the number of side effects, I think it's really important that we consider sex and gender-specific factors when we do this type of research. But having said this, sex is really not everything. So we have to keep in mind that even though we see sex differences for almost everything we look at in the brain, these are really small, and they're likely completely meaningless for you on an individual basis. So I won't be able to tell anything about you just based on your gender. This is not a useful information. Just on a group level, it's a useful information when we do research. So I don't really think there's a need for researchers to specifically look for sex differences. Instead, I think maybe the approach we should do is that we should always start with a representative population of whatever we study. So in mice, this will be male and female, but if you do humans, they should obviously include people from different socioeconomic backgrounds and ethical backgrounds, different ages, and then we can examine these differences, what they mean. So if we don't find any differences, so if we don't see any sex differences, then we normally go on to just pool all the animals and use this as a representative group without further considering sex. And if we do see sex differences, this is when we split the groups into two and start specifically examining why we see these sex differences. So what's the biological reason behind this sex difference? And in most cases in the lab, we don't actually see a sex difference. So it's not that it exists for absolutely everything we look at. But of course, given the topic of today, Tobias will now tell you about an example from his work studying depression, where the sex of the animal really did matter, and where we would have missed some extremely important information if we had just used one sex. So we'll hand over to Tobias then.
Yeah, thanks, Hanna, for the introduction. And also, again, my name is Tobias, and I'm a postdoc in the lab in Buch at MDC. And my research is looking at how stress can lead to depressive behavior in animals, and then what's the molecular background of that. And here you can, I think you can see it. That's our setup where we do where the animals live during the day and night for their whole experiment. And here, I think that's 2 females that we can see here. So I'm also looking at males and females in that study. But what was really interesting and also important for me was the fact that in human studies and human research, we know that that not everybody who has a stressful event during their life will become depressed eventually. It's just a subset of people that develop depression after stressful life events, and stress is one of the major factors to develop depression or any psychiatric disorder. And we also then wanted to ask that, how does it look in animal models? Do mice also have this trend that not every mouse that is being stressed is getting depressed? And for this, we set up this hypothesis, or actually it's in the literature also an hypothesis, but we set out to really look at that specifically, meaning that if we stress our mice, we will have animals that might become depressed and others that might stay resilient towards it. And here we then also wanted to include, as mentioned earlier, how sex as a biological factor is actually influencing the outcomes or the phenotypes, the symptoms that those animals have later on. And the stressor, as Hanna also mentioned, is during an adolescent phase. It's a phase where the animals are still undergoing brain development, so all your neural circuits will shape during that time, and stress can really positively and negatively affect this. So coming back to what Hanna said previously, or showed this figure here, how you assess usually depression in humans. There are different scales. This is one scale. And when then looking at animals, how can we study those those symptoms in animals? Or can we actually do that? And yes, we can. At least some of them. I mean, we cannot ask the animals if they have suicidal thoughts. Maybe if there's a mouse whisperer here among us, but yeah. But we can actually, and that's also where then the science comes in, we can measure. And we can measure, for example, anhedonia, which I think was also shown on the word cloud, which is like the loss of pleasure or loss of interest and usually pleasurable things, like watching TV, playing, or like going out with friends. So this is anhedonia. And we can also measure that in our rodents, in our mice, meaning they love sugar. And one way to test if they lost pleasure is, do they still like sugar, for example? Then we can also measure their social interaction. Are they interested in social interactions, or is the sociability changed? We can also look at differences in their general day-to-day life in the cage. I mean, it's not that much to do in the cage, but they can run around and they can sleep and they can interact with each other. And this is what we can also measure. Besides other more classical tests here, for example, we can test the memory, cognition, which is also important, as we heard earlier, in depression, since cognition or memory is usually affected badly. And we can also actually measure the stress hormones from the blood, which you can also do in humans, interestingly. But yeah, so how do we then actually measure this behavior in animal models here? And for this, one thing we set out to do was to automate our behavioral analysis, since we have quite a lot of animals, and we also want to have the assessment of behaviors more or less standardized. And for this, I don't know if you can see that, but the animal, I think the color is not the best. But yeah, we can automatically track those animals and then also analyze. For example, in this case, this is the open field test, and open fields mice don't like it because predators come from above and being in an open area is dangerous. So a mouse that is more anxious, for example, would stick more to the walls here, whereas a mouse that is less anxious would also spend more time in the center of this arena because they love to explore. Then a next test here, this is our memory test, where they are given 2 objects, and we measure how much time they actually spend investigating them. Since also novelty, they like novelty, they would prefer the novel object. And here, this is 2 times the same object, and now you just have to imagine that we switch out one object with one that looks completely different, and then we ask the mouse, which one do you prefer? And usually they should prefer the new object if they can remember it. So if the memory is also affected, we would see that here, in that they don't have a preference anymore for the novel. Another test we do here, also to measure anxiety, this is called the elevated plus maze, which is basically, it's high, and it has 2 open arms and 2 closed arms, and yeah, it's scary for the mice to be on the open arms, so they, if they're more anxious, would also spend more time like this guy there in the closed arms. So right now, he's looking down, this is very brave, but then he's deciding, okay, no, I don't like it. I go back to safety. So this is what we can do, and this is just a selection, but this is what we can do in the lab to study those relatable tasks or feelings of those animals. And since we are also interested in to assess how stressor exposure during adolescence is affecting those types of behaviors, we then turn to a stress paradigm. Because, as I mentioned, stress is one of the major causes for psychiatric disorders in general. And we turned to the social instability stress, which is relatively simple. We have our mice, they live in groups of 4 in a cage, and we just switch the constellation of those cages 2 times per week. Imagine if you always, like every week you're in a new social group, you have to adapt, you don't know the people, you're like, okay, who can I trust, should I mistrust or anything? It's also stressful for them because they need this predictability, and if they have a social unpredictable environment, that's scary and that's stressful. So this is why we also chose the test here, because it's very relatable also to the human cause of stress. And in this cage, they are running day and night, and you probably wondered what those dotted lines are below. This is our radio frequency antenna with which we can pick up the movement of the animals through the whole time. They get an antenna implanted under the skin relatively early, and then they are put into this stress paradigm for 2 weeks during adolescence. So this is at around from 3 weeks on to 5 weeks, they are adolescent. And we wanted to really then see how this stressor during this vulnerable time period for brain development can actually affect their behaviors. After the test, they remain in the last social group, and we also then tested all the classical behaviors. And as you also remember from earlier, we also wanted to look at resilience or susceptibility or vulnerability factors in those animals. But how do we actually decide how a mouse is vulnerable or which mouse is resilient to the stressor? And for this, we got an inspiration also from human psychology, where usually you need to have several symptoms present. So it's not just, okay, you have a bad day for one day, you must be depressed. No, you have to have it for a longer time period. And you also have to have more factors than just one. And for this, we chose our anhedonia test, the performance of the mice, how they perform. Do they like sugar? Did they lose the liking of sugar? Then how is their cognition? Do they have memory deficits? How is their general weight development, which is also an indicator for food intake or like diminished food intake? And then we are also measuring other weights, for example, here the spleen weight and adrenal gland weight, which are also known to be linked to depression in humans and adrenal gland weight, because it's the main the main location for the synthesis of stress hormones, which we can also measure. In this case, mice have corticosterone. Humans usually have cortisol. I don't know if you heard about that, but this is the main thing that's important for the stress responses and also important for shutting down the stress responses again. But I won't go too much into detail. But yeah, we take those 6 factors and group them for each individual together so that we have one score for each individual, and then we compare that to our controls. And this is how we envisioned it, how it might look. Where we have on the left side our control animals, they have a normal performance because they're controls, and then we will have our stress group here on the right side, where many will have similar performance like controls, but we also have some that actually perform overall worse than our control animals. And these are the ones that we then, once we split the stress group, we then termed as susceptible or resilient. And looking into our data, which we have so far, is that indeed we can, I don't know if it's loading, indeed we can see this difference between the resilient and the susceptible animals here based on the 6 different parameters I just showed you. And here we see that roughly 50% in both males and females are actually performing significantly worse than the control animals. Still, we have a huge population that is relatively normal, but we have those that actually are more affected, which we wanted to have in the end. And what we're then looking at further is where do they differ, the males and females in our experiments. So since it's important to always look at, different sexes, we looked at, for example here, physiology, where both males and females are affected to some extent if they're susceptible. But then interestingly, mainly on our depressive and cognitive realm, we have mainly males that are negatively affected if they're more susceptible towards the stress. We also saw similar differences then in the home cage testing, which I will go through with you now. Our home cage, as you already saw a bit earlier, it looks like this. It has, on the left side, it has a water bottle. On the right side, it has a little feeder for food. I mean, those are empty now, but this is usually how our setup looks like. And in there, we can measure different things. For example, we can measure their baseline activity. Since activity also in human psychiatry is also thought to be linked to psychiatric disorders, and also in humans we can, it's actually I think also tracked here at the Charité with like little wristbands to just measure their activity, how much steps do you get in your day. And this is something similar here we can also assess in rodents. And what you see here is a curve for, it's like 4 days with the night phase and the day phase. They are nocturnal animals, so they're active during the night, and they are inactive, so they sleep during the day. And this is also what we see here. One interesting thing is actually what we saw more in younger animals is that they really like to take an afternoon nap. I think like little kids, they are active in the mornings and midday, and then in the afternoon or actually also humans have that, I guess, that in the afternoon after lunch you really feel like taking a nap. And we actually also see that in our mice here. It's kind of nice to see, since we also didn't expect that. But when we're then looking at our subjects, first in the males, if that comes up, yeah. First in the males, when we're looking at the activity overall, it looks relatively similar. We have our control animals here in dark blue and our susceptible animals in orange. And here we see that during the inactive phase, so when they should be sleeping, this is the day. It's a bit confusing, I know. But we see here when they should be sleeping that the males are more active, so they travel more when they're susceptible. In females, we see the same, that they're also more active, but also on the same, they also sleep less, or they tend to sleep less compared to the control or the resilient females. So here we have actually also then a similar diagnosis as also in humans with depression, that they also have sleep problems. And this seems to be also true in our females here in this case, that they have disrupted, shorter sleep. Then another thing that's usually done in stress research is you present them with a stressful challenge and then you measure how do they react to the stressor. And usually that's done outside of the home cage in some artificial area. I mean, this is also artificial in the home cage, but in a more stressful environment. And here we also wanted to implement that into our home cage to really see how they behave in home cage conditions. And for that we put a little cookie into I think, who knows Tresor? Kellogg's Tresor? Yeah. They really love it because it's sweet, it's sugar, it's nutrient rich, and they fight for it because they love it. They ignore their standard food, so it's like also interesting to see. Yeah, it's a high reward food for them. And when we then look at the activity curves, we see that males and females on the left and the right, they show higher activity levels. But only on the left side, the males that are susceptible to the stress are actually more active than compared to controls and also compared to the resilient animals. Whereas in females, it's more both, regardless if they're resilient or susceptible, they're both more active, which is a nice indicator also for the stress response. So higher stress response means that there is some dysregulation of this stress response in the brain and in the body, which then also can lead to the resilience or susceptible phenotypes we see. So here also we see differences between sexes, although both respond. The males, the susceptible animals, respond stronger towards this stimulus. We also have other home cage challenges that I didn't show here, where there also are differences, but this is really nice to see how they are actually also different in the behavior. So since this was all now behavioral, I won't go too much detail into what's next, but I will just mention it a little bit, because since our group is called also the molecular and cellular underlyings of behavior, we are also now interested in, okay, what's actually different on a molecular cellular level. And for this, we zoom into this brain. So this is a brain of a mouse. When you cut through it, you can stain for proteins, for different proteins, and then you can select single cells to then really analyze on the single cellular level the differences between our susceptible or resilient animals, to try to find a mechanism that is underlying or potentially underlying the differences we see on the behavioral level. So, and with this then, as a summary for now, just very quick, sex differences are important and they influence how stress is affecting behavioral and physiological parameters or outcomes. So yeah, with this then I think the take-home messages.
So I hope we really convinced you today that it's really essential to consider sex and gender, but especially sex then if you do animal research, as a biological factor when you study the brain. But also I want to leave you with a take-home message that sex differences in the brain doesn't really define you at an individual level. So instead of specifically focusing on sex difference, all of it's incredibly important we take it into consideration, we should focus on using a representative population for whatever it is we study, so that we can move to a more personalized approach to medicine that takes sex and gender into account, rather than sort of a one-size-fits-all approach that's traditionally been the way it's been done. So with that, I want to thank all of you for your attention. Also really want to thank my fantastic team, so including Tobias, so thank you so much for doing this with me. And we have 2 team members here today, so Rosalba and Clara. I want to wave. Very grateful to have them in the lab. And we will now move on to a Q&A. And so Tobias is likely German, so if you don't feel comfortable asking a question in English, you can ask it in German and we will translate it.
Are there already some people who want to ask a question? Otherwise, I will be. Oh yeah, already. Wonderful. Geht gleich los.
First of all, thank you so much for this amazing talk. Just because it was the last thing and it was the first question that popped into my mind, you said that it's important to study representative samples, whatever you study. So what does a representative sample mean with regards to mice, I wonder?
Yeah, so that's a good question. The mice we use are generally genetically similar. So there's different strains of mice, and most scientists use one strain, and this is for practical reasons. So we should think about what's called a 3R. So we should limit the number of mice we use for our study. So if you would use mice from lots of different strains, you would need more mice, and it would also increase the variability of your research. So the thought is that if we use one strain that is really similar, then we can focus only on the differences we're interested in. So in our case, we can kind of ignore a little bit the genetic differences and really only focus on sex and age. So generally the other things that it's important when you study mice then is, so sex is the most obvious, use both sexes, but also use a different age span. So of course in this research our question was really what happens if you're stressed during a very specific time period. But for example, in other part of the lab, we use mouse models for autism, and there a lot of research is done just looking at adult mice, which is not at all representative. So autism is a lifelong condition. So there, ideally, for example, a better representative sample would be to look at young mice to old mice for a longer time span. So, like ideally, we would use like very genetically diverse populations of mice through different ages and both sexes, but this is not always possible for practical reasons.
You showed these bloods at the end, and we saw that they differ quite a bit. Right? You saw some significant differences between the sexes, but still they differed quite a bit. And when you then say that the difference is anyway rather small between the sexes, I want to ask the question if at some point we come into that area where we say it's just individuality and not sex. Would that be possible, or would you say no, still it is clearly a sex difference here?
Yeah, I think that's a bit difficult to answer. Of course, individual differences are important, and I think there. It's on. There are some movements, especially in depressive research, to test, for example, for some known genetic factors. For example, for SSRI, serotonin reuptake inhibitors, there are some genetic underlying causes that are known for people that might hint that they might not work for them. But like in general, for individual differences, yes. I mean, this is the idea of personalized medicine, right? It's a bit difficult because, I mean, for certain, I think it's also done for cancer therapies where you can really personalize this medicine. Ideally, it would be ideal, perfect. But the other thing is it's difficult to do that for everyone, and you want to have a treatment for every demographic and everyone in a way, so you have to find some common grounds, some common mechanisms that you might then treat more in a broader approach.
Also, a question to you, Tobias, since your university times are not so long ago as ours. Have you been taught anything about sex differences in the university, in any respect?
I don't know. I think it.
Have you, Hanna?
I'm pretty sure not.
I think it depends on. I mean, yes, depends on in lectures, not so much. I mean, of course, you have also sex there, but it really depends where you then do your further practical courses or lectures. For example, I did mine at in psychiatry, and that was important to also recognize.
It's funny, but I mean, you studied biology. I studied biology too, and it's funny now that I think about it, that such a crucial biological influence factor is completely diminished in physiology. Hanna, when did that factor come in? No.
For me,
yeah, you know what I mean.
well, so I've always been very politically interested, so I've always been a feminist and always found women's issues personally very important. So it's always been something I've been interested in through the back of my mind. And in research, it's also something I've always studied when I could. So in my PhD, we did not do it. It's incredibly difficult to sex fish embryos. But as soon as we could do it, we always take it into account. And there also, I mean, what you said with the personalized medicine versus sex differences, because this is, of course, always something to keep in mind, and why I've emphasized so much that the differences between sexes are small. Because studying sex difference have also then historically been used as an argument that women are worse. And it's very important to notice this is not the case. But because, like Tobias said, we can't, for practical reason, we can't go to every one of you when you come in with a depression diagnosis and start from scratch. This is not possible. So we have to try to divide people into groups and try to understand that, okay, if you're part of this group, whatever that group might be, then maybe you're more likely to respond to this treatment. So you might be more likely to get help from this than from something else. And this, will then hopefully we can, as we learn more and more and more, divide these groups into smaller and smaller groups. Hopefully we can get some biomarkers so we can test your blood maybe, and this will give us a clue. But because we know that sex is such a big factor in this, because we can see this in the diagnostic data and in the symptom data and also treatment data. So it is, I think, one of the really kind of obvious things to start with.
Thank you for your talk, both of you. And I was actually wondering, it was curiosity. Like, so you said that the male mice develop more depression compared to female, and this is a big difference with humans being that you show instead that it is the reverse. Do you know at the molecular level what is the difference? Why there is this difference? What is the impact? Because we are not mice, the three are ethics, we know. And how you can solve this problem, how you can explain this difference at the molecular level, if you can. And then I have a second question, if I can.
Yeah, I mean, that comes then down to, I mean, I don't know why males here are more depressive or more symptoms, but that's also what we want to look at in the end, why we have some differences there. But on a molecular level, I mean, you have hormonal factors. So as you know, there's not just like two hormones. I don't know, oxytocin or I don't know, other hormones, acetylcholine. But we also have sex hormones, for example, and sex hormone receptors, both in males and females, or men and women as well. And responses to stress can also be dependent on how you react to, for example, different hormones or what your hormonal level is at the moment, not just in women, but also in men, because men can also have fluctuations in hormone levels depending on their mood, depending on the state, and depending on the environment they're in. So this might be a confounding factor when looking at differences between sexes, that they just respond differently with their current hormonal status.
So also if I can add, so we have quite good reasons to believe that things we find in mice at least have a relatively good chance of being translatable to human. And one of these reasons is that, for example, if we give mice antidepressants, they do better. So they respond similar to humans. And if we change the expression of genes in mice that we know are linked to depression in humans, they will also show this type of depressive-like symptoms. And a lot of other things also translate. So changes in brain level, changes in gene levels in mice in the models we have for depression. So at least some of the biology is likely to be the same. Obviously, we can't be sure. But we certainly hope that, we think there's a good chance that at least some of it will be able to be translatable.
I don't forget you, but I first let others speak and then get back to you.
Yeah, thank you very much for the talk. I just want you to comment on difficulty how to interpret animal behavior. Because, for example, you show that example with open field, and you say it stays on the side, so it's more anxious. It goes to the center, maybe it's more brave, but you can also interpret it as like his cognitive abilities are not right. That's why it goes to the middle, where it's maybe dangerous. So how does it influence your decisions? Like, how do you interpret it? How difficult it is to translate this behavior to human behavior then? Thank you.
Yeah, I mean, there, what Hanna also just mentioned is that you can test this with, for example, anxiolytics. If you give animals anxiolytics, they will spend more time in the center of the arena, for example, which is thought that it's therefore anxiety that they have at that point, so that they're more anxious. Because same with antidepressants, antidepressant treatment can also improve some of the experiments or the symptoms in those experiments in animals. But of course, we cannot be sure. We can just observe it in this case. And this is also a thing, in especially neuroscience, it's done a lot that you overinterpret things, right? But in this case, since you can test it with actually medications that also work in humans, we can say, okay, it's most probably anxiety that they have in that moment when they are spending more time on the walls.
Thank you. I was wondering if you controlled for menstrual cycle in females when you stratified in susceptible and responders?
In general, it's kind of hard to do that every day. So no, simple answer, no at the beginning. But we do it at the end of the experiment. So when we also take a blood draw, we also look at their current estrus stage. But of course, it's not that easily to implement if you have it's a lot of animals. But that shouldn't be the excuse, by the way. But the more important factor is that, for example, in humans, when you give treatment, you also want to have, in the end, treatment or symptoms that are not just present during a very specific stage of the estrus cycle. So that's why we also chose to not completely integrate that.
So mice cycle roughly every 3 days, right? So they should go through multiple cycles during the phase of this experiment. So it might impact specific behaviors, but we should overall capture changes because we look at them for so long.
One more here at the side.
Well, thank you very much for this very nice talk, and also to point at this very important issue, I think. I have a question regarding the type of stress. Does it have an impact how you stress the mice, on how much the sex has an impact, like whether you have a very short and shocking stress, or whether you have another type like grief or famine or something like that?
Yeah, definitely. I mean, not every stressor is the same, and the animals also won't respond to every stressor in the same way. So there's a lot of variability there. Some stressors might evoke more anxious behaviors, others more depressive behaviors. I mean, here we didn't see any difference in anxiety, for example. With other stressors, you might actually find more anxiety or more depressive. But yeah, the stressor matters, and also how strong it is, and also how long it is in the end.
But so this is also generally not known in people. So if, for example, different types of stress will activate different pathways, we don't really know this. And we also don't really know it in mice. So even if they show different symptoms, is it kind of roughly the same brain areas that are involved, no matter what in the end? And this is something we're actively interested to pursuing in the lab. And we also, so Clara, who is here, she also has an extremely exciting project where she's looking specifically at shorter stress in female mice and how this impact them. Also then related to their hormonal cycle. So
I saw one arm here on the side somewhere.
Yes, thank you. I was wondering about the statistics you showed earlier, that women have more depression than men. Is it can it be biased because women tend to go more often to the doctors, and this is why it's more easily recognized? How would you evaluate this statistic?
So it's extremely likely that that's a major factor. So, I mean, I don't have good evidence for this, but I personally don't believe that there is an actual twofold difference. It's known that women are generally speaking more likely to seek help and men are less likely, so that's also why they're kind of more outward acting. The symptoms might also be different in ways that means that it's not recognized in depression in men as often. So they might, even if they seek help, they might not get diagnosed, for example. It might also be more, might be more socially acceptable for women to seek help. So all of this will really matter, and I think this plays a really big role in the prevalence. I still think probably there is a biological difference, like as a group, that impacts this. But I think in terms of prevalence, probably the societal factor is the biggest part driving it.
Thank you for your presentation. I have two questions. One very short. When you are mixing the groups in the social setting, they are mixing with the same sex or the different sex as well?
Same sex only.
Same sex only. So okay. The second question is the important question. I realized that when you're increasing the stress in the adolescence of the especially male mice, when they grow up and became adolescents, there is a performance difference between them. So some of them are performing less than the control group, and some of them are performing actually better than the control group. So this is a wild question, though. Can we assume that in the later stages of these mice, can we observe more social inequality in their reaching the mates or reaching the resources if they are stressed in their adolescences?
Yeah, that's an interesting question, actually. It goes down to this theory that is also there with the mismatch hypothesis. For example, if you have a stressful childhood, that you will technically cope better in a stressful adulthood life, which would be a match, so you would not have negative outcomes. Of course, it might be that those animals that are then resilient are also more successful in later life. We didn't test that, but that's a nice theory. But yeah, that wasn't really the focus of the study, but it's still interesting to think about. Yeah.
Hi. You were talking before about the environmental things that affect depression, and something that comes to mind for women is obviously birth control, that females have to take more than men. I would want to ask what depth of research has been done to the effects of birth control on mental health, as this is clearly something that females have to do more than men.
So the short answer is very little. So it's known, if you just ask women, if you would poll your friends on birth control, people will tell you that it affects your mood, and that it affects your mood in different ways, right? We know this, but how it actually interacts with depression is really poorly studied, and if you get in too much about it, you can become very angry. But the research that is there doesn't necessarily go in the way that people might think. So, for example, a recent study suggested that in people that had been diagnosed with depression, it was better for them to, and was on birth control, it was better for them to stay on the birth control, so not take this 7-day break, because they generally felt worse during this break. So hormones might work in both kind of smoothing out your mood, so you have less kind of ups and downs that might help you in depression. But it might also, of course, influence it in a worse way, and probably this is going to be very different for different people. So for some, it might have a negative effect, for others a positive effect. It might also be different for different types of birth controls. So you could be on progesterone mostly, or estrogen, or mix, or how you get it, how long you've been on it for. So likely this will be very personal, but you would need a lot of more research for this. And hopefully, maybe in a few years, we can do this again, and then Clara can talk about it, because she will look specifically at how birth control in mice affects their response to stress.
I want to come back to you. Your second question. Uppsala.
I'm just a curiosity, because there are several studies on depression on the gut-brain axis, how the microbiome influence depression, right? So I was curious if you never try to give different food, high like fast food, like whatever, like fat or light food, if influence somehow the behavior of the mice after stress or without stress, and gender dependent, the sex dependency.
Yeah, there are studies on that. For example, with Western diet, right? High carbs, high fat, and it influences behavior negatively. So they should tend to show more depressive-like behaviors and anxiety, besides also being overweight, which is also a huge risk factor. But that then goes into inflammation theory and everything. So in general, what you eat is also influencing how you feel and how you are living your life and every day.
Just as a promotion then, I think, so one of my colleagues, Rachel Lippert, she researched this and she did one of these talks earlier this year, I think, so it should be on YouTube. So you can look it up and then learn much more about it. Because, yeah, I mean, we didn't do it, and one of the reasons is you would need an extremely large cohort of mice, so we haven't had the time.
Hi. Hi. My question is, you described depression as episodic, and I wanted to know if that's the same for mice. Do they also have an episodic character? And how would they cope with it naturally? And is there something we can learn from them for human behavior or treatment?
I don't know if so. First, usually when you look at mice depressive-like behaviors, you look at it once for 1 week, for example, with all the behaviors, and then you stop the experiment. I mean, there are some experiments that are also aiming to go more in the long term. For example, they test the animals and then they retest them 1 month later. But there it's then more going into the direction, okay, how chronic is this behavioral change? I don't know if there's any study that is also looking at this episodic phenotypes. Since also mice, they are not depressed, they just have these symptoms. So, but yeah, I mean, might be also interesting if of course, if there's an episodic factor to that, but it's kind of hard to study, I think, in general.
So we could, I guess, see it in the home cages if we had them for long enough. I mean, this is back to this question about how you use a representative population in mice. Generally, this is an issue that people study behavior for a very short period of time, and then you get this snapshot. It would be much better if we could really follow them for a long time. So there's a couple of reasons people don't do that. I mean, one is cost and time, obviously. So, for example, we want to do start a project on aging now. It takes 3 years. Because that's how long the mice live, and a PhD is 3 years. So it's very risky and difficult to finish it on time. But also it has been very difficult because you would have to test the mice over and over and over again, which is stressful for the mice and very time consuming. So these cages that Tobias talked about, the home cages, they're relatively new technologies. So I think now is really the time to do this type of research. So now we would be able to look at it and also then be able to really pick out kind of when episodes happens, what they're linked to. So yeah, something you can think about for the future.
Yeah. One more question here from the side.
Hey, thanks for the talk. Curious about the fact that like humans, mice are social beings, and like when you do the experimentation in groups of them, and I was wondering if there's some influence of the group in the individual. For instance, if you could select a specific individual resilient and put into a depressed group, what would be the behavior? Or the opposite as well, you put a depressed into a resilient, if it could develop some resiliency somehow, and if there are any aspects of sex difference in these groups. Could that study be made? Is it possible or not? Or
so you mean if we set one susceptible animals in a group of resilience, that they would that the susceptible would become? Yeah, I mean that's an interesting question. How interactions between them also influence the behaviors and or the susceptibility. It's probably possible to do that. It's just going to be difficult.
I mean, we can partially do it already with our data, right? And it's on our list. It just requires quite a lot of modeling. So, because we have information from every single animals, and we know which cage they were and with which mice they were at which given time. So we can, for example, test if one mouse is always social, no matter like who it hangs out with, or if it's kind of specific. So if it likes some mice but not other mice, and how maybe this might impact it. So maybe you have like hotspots. Maybe you have one mouse that makes everyone else feel bad because it's a terrible bully. And so we can look into this in the data. It's just because of the lot of cage changes, we need to do quite a bit of modeling, and we haven't gotten around to this yet. So a lot of this data is very, very new, but yeah, we can partially maybe answer it.
And technically also every individual should be in the cage with that one bully mouse, for example, because we're switching them around so everybody meets everybody at least once.
The core of the question is like, could depression be influenced on others? Like, do you think?
I mean, there are. I mean, of course, your general mood, can also influence the mood of the people around you. That's whether you're healthy or if you're depressed. So, I mean, there's also this terminology in psychiatry of this. I don't know the terminology specifically at the moment, but where you can also be like codependent, or like you can also develop, for example, if your spouse is depressed, you also have a higher likelihood of also developing some of the symptoms, or also depression, yes. But this is not specific for depression. This is also when you're in a good mood, it's also more likely that your closer circle will also feel better then, because they're more influenced by you.
And when having a social circle, a large social circle, and being socially active is protective generally for depression. So it's protective for lots of things. It's also protective for Alzheimer's or lots of other disorders. So one of the best things you can do if you want to stay healthy is be social and meet lots of people. And this is actually also the same for mice. So if we single house mice, especially females, they really don't like it. So they start also showing quite negative health consequences. So having kind of a social environment is also really important for their general well-being, same as for us.
Thank you very much for the talk. Just one question. So as far as I understood you, is that now we see those differences that sex can make in the development of depression, but we don't really understand yet why that's happening, right? Can you comment a little bit more if you have already seen anything in the structure of the brain, as you showed the last slide, which was showing like this microscopic view of the brain? Is there any differences that you can see that you can link to sex?
No, I didn't look at that yet. I mean, in general, that most likely comes down to the differences in sex hormones and the receptors, because that's the main differences, the difference that we can measure there. I mean, of course, we can also measure differences in volume, brain volume, hippocampus, which is important for memory volume. But on a morphological level, I haven't looked at that, no.
So what we will look at, and again, this is very new data. So this, what Tobias showed, is what he's doing now. So what he's doing is he's with a laser cutting out specific neurons, which is very time consuming, and then he pulls them, and then we do a technique called mass spectrometry or proteomics, where you can look at not all proteins, but a lot of proteins in your sample that we can then link to the behavior of the individual animal. And we have done this in other projects in the lab, so where we just look at sex differences generally in the brain of male and female mice. Also just looked at specifically at proteins that are located at the synapse. And there's very, very few differences at baseline. So generally speaking, the brains of mice they're quite similar on the kind of protein levels at baseline. So if we stress them or do different things to them, you start seeing differences. But we can for sure look at it, and yeah, hopefully in maybe a year's time, we will know at least some of the answer to it.
One more question. Thank you. My question is about possible sex differences, if any, about what is called stress-related growth. I understand that there are several studies in behavioral sciences that demonstratively prove that there are some people who are able to actually grow as a direct result of experiencing stress, whereas other people experience detrimental effects. There are these people who actually grow, which is why which led behavioral scientists to coin a new term called stress-related growth. And this is not just about stress, but all sorts of challenging situations, including traumatic experiences. So.
So when you mean growth, so like I guess not becoming larger, but
not
grow as a
physical
person.
Growth, not physical growth.
Yeah. So this is very likely. I mean, you see it in, like Tobias said, that you have some people that experience stress will then later on be better at coping with stress because your brain has this experience, it knows how to handle it, so the next time it happens, it will do better. But as for people who we call them vulnerable, they will experience stress maybe once, and instead of learning how to cope with it, they will do the opposite. So the next time it happens, their brain will maybe over-respond, and they will instead do worse. And in people, it depends a little bit, of course, on the stressor in the study, but actually most people who experience even extremely traumatic events will be totally fine. So most people will not experience depression after something very traumatic, but some people do, so roughly around 25%. And this is then similar to what we see in our animal models. So only some people or some mice will show these type of symptoms after a stressor, and others will not. And some of them, we didn't really show this data, but some of them actually seem to maybe perform a little bit better in certain tasks. And we're also then looking at the brain not only of the mice that shows depressive-like symptoms, but also the mice that are resilient because one thought is that we spend all this time looking at what makes someone depressed, but it might be more useful to look at what makes people resilient, because that might be a way to identify new treatments that you otherwise will not be able to tell, since these people obviously have something in them or that makes them kind of naturally resilient to depression. So that's what we eventually will know, hopefully.
Thank you. I see there are many more arms being raised, and you can all ask your questions, but the problem is we have to shut down the event officially. So I would just put this to an end, but you can all afterwards still come to us and ask us and discuss with us. I just wanted to, for us all, thank again for this wonderful presentation of Tobias and Hanna. Keep on applauding for Felix at the camera and Susie at the technological stuff. Thank you all for being here today, and I'm looking forward to see you all again next year. We'll continue with Berlin Brains. Find more info on www.urania.com. And of course, we will see each other tomorrow and the day after. Tomorrow here in the planetarium, one floor up with Einstein in the dome, and the day afterwards in the Roadrunner's Paradise for the Science Slam of the 7 Excellence Clusters. See you then. Have a good night. Thank you all for being here. Bye.